First of all, I would like to thank the organizers for inviting me to give a presentation as part of the COVID-19 Vaccine Development and Implementation Workshop 2021 webinar. My talk today is going to focus squarely on an update on current vaccine situation and overview. This will serve as a primer for what should be excellent talks throughout this workshop. My disclosures are listed. My research program has relationships with pharmaceutical groups that are involved in the COVID-19 vaccine development process, none of which would influence my presentation today. For this talk, to provide you the most up-to-date information, I have added data from press releases and preprint servers. However, I have prioritized published data above any of these two. Our outline today will focus on the following. We'll talk about some of the lessons learned from the role of vaccinations in contemporary epidemics. We will discuss the current clinical trials landscapes, lessons learned, and future directions. We'll discuss the vaccination platforms, pros and cons, and I'll give you a 10,000 feet overview of the preclinical studies, safety, immunogenicity, and efficacy data that have emerged from the contemporary clinical trials. And lastly, just a few slides on the vaccine rollout. I wanted to start by highlighting some very important quotes that I have come across that I think convey strong messages to the audience. The first is a comment by an author in the Frontiers of Immunology, highlighting the public health importance of vaccination as being the most successful medical approach to disease prevention and control. And I wanted to also mention a quote by Anthony Fauci, who, when the phase 3 trials were up and running, mentioned that if we get both of these together, an effective vaccine and public health measures, that would really be a game changer for the COVID-19 pandemic. And lastly, a comment by Jonas Salk, a reply when he was questioned on the safety of the polio vaccine he developed. He said, "It is safe, and you can't get safer than safe." So we'll discuss the history of vaccination epidemics in this slide, but really just wanted to highlight some lessons from the Ebola experience on how vaccines can play a great role in addressing a current epidemic or pandemic. So some of the lessons learned from the recent West African 2014 to 2016 Ebola epidemic that did spread to quite a few countries, even outside the continent, we learned that innovative and accelerated clinical trials that don't compromise safety can be conducted, as has occurred with COVID-19. We've learned that epidemics or pandemics with rapid clinical courses have an advantage. They allow for rapid evaluation of therapies and vaccines, again, as has occurred with this pandemic. We know that it is very critical that global community collaboration by academic institutions, industry, as well as regulators are critical to provide a rapid response to curb some of these epidemics. And lastly, and no less important, is the need to work with local communities and stakeholders who would receive these vaccines to make sure that they're willing to embrace vaccines, especially when they're novel technologies. Now, this is a pandemic vaccine model. On the top part of the figure is the traditional vaccine development model. I think most of us know that the earliest timeline from initiating studying a vaccine to having it approved for use in humans was about 4 years for the mumps vaccine. And these were typically done in sequential stages, preclinical development, clinical development, and licensure, after which production was initiated. But as you can imagine, because of the urgent need to develop a vaccine for COVID-19, there's had to be an accelerated model that has had the ultimate goal of shortening the vaccine development time. And this has included overlapping phases from phase 1 through phase 2 and 3, working with regulators to review data quickly to make decisions on emergency use authorization and approval, as well as initiation of production, having seen an early signal from early phase trials that suggest that a vaccine candidate was promising, along with all the resources that are required to have that done. This model has definitely showed us a quick and efficient way to do things, and I hope it serves us better for current and future infectious diseases for which vaccines are being developed. However, as you can imagine, one limitation to this accelerated process is that you may not have long-term safety data when approval and licensure decisions are made. The vaccine development process has led to key outputs, and I know most of us are very cognizant with the different phases of vaccine development, but permit me to make a few points. I can't highlight how critical it has been for preclinical studies to inform studies in humans, establishing safety, identifying the appropriate and best vaccine candidates to be advanced to human trials. The animal studies have also allowed for viral challenge studies to have that early assessment on if these vaccines can protect against disease. The phase 1 trials have provided very important safety data and have allowed us to calibrate very carefully the doses according to their side effects without sacrificing immunogenicity. The phase 2 studies have also provided critical data on immunogenicity as well as its performance in patients with different characteristics. And we all know and have celebrated a lot of the success with the phase 3 trials that have provided data on large-scale efficacy and safety. But that's not the end of the story, because we know that as the vaccine is rolled out, post-approval studies and evaluations are ongoing, which are no less important for us to understand, for example, much greater detail, the safety signals for these vaccines. For example, there's a vaccine adverse event reporting system that the CDC and FDA run in the United States that allows us to capture with the vaccination rollout side effects or adverse events as they occur and keep a log of those. Some studies that will be needed will be comparative studies in the future to allow evaluating either different vaccination strategies or comparing vaccine candidates, as well as expanding study protocols to study populations that were left out of the initial phase 3 clinical trial protocols. We're still expecting a lot of important data from the phase 2 and 3 clinical trials to provide information, for example, about the durability of the immune responses and also to continue to access long-term safety signals. We've already talked about the vaccine adverse event reporting system that will provide safety data. And the registries are being established and could be established to continue to assess surveillance for safety events among unique populations such as pregnant women, other immune compromised patients getting vaccines, and may even provide some evidence of efficacy in these subgroups that were not included in the initial clinical trials. We know that there are new variants circulating and new batches of vaccines to be created to address those. Is yet to be seen what the regulatory process would be and how rigorous the expectation would be for these new vaccines. And there will be a talk addressing some of these regulatory issues around the vaccine development process. This is the COVID vaccine clinical trials landscape, which I think many of us are aware of. This is really updated daily by the New York Times. And here you can see really a flurry of activity of multiple clinical trials that are in different phases of development, the minority of which have achieved authorization and a few that have been approved, as well as those have been abandoned for varying reasons. We do know that this is good news because we need as many effective vaccines as possible to meet the global demand and to really have that, achieve that elusive goal of herd immunity on a global scale. Towards the right, you have the figure that shows the lead vaccine candidates, their vaccine platforms, which we will discuss in subsequent slides, the phases that are currently ongoing, and their authorization or approval process. This has unmasked the fact that there are variable regulatory standards and approaches in different parts of the world that are not necessarily harmonized. And also to just mention that it's been heartening to see that the lead vaccine candidates, and lead by lead, I mean vaccine candidates that have reported on their phase 3 results have been the newish technologies like messenger RNA technology and viral vectors, which have really opened possibilities for addressing other infectious diseases in the future. This is a cartoon of the vaccine approaches for SARS-CoV-2 virus, and this is an image from the frontiers of immunology. This just goes to show that multiple approaches are being evaluated for vaccines for SARS-CoV-2. In brief, there are nucleic acid-based or gene-based strategies, which can be either mRNA or DNA vaccines. There are viral vector technologies, which include both non-replicating viral vectors and replicating forms. They're whole virus vaccines that can either be live attenuated or inactivated or viruses, protein and peptide subunit viruses, nanoparticle and virus-like particle vaccines, as well as immune cell therapy to address the virus. However, we do know as these approaches are evaluated in clinical trials, the key considerations would be how immunogenic they are, how safe, effective, what kind of delivery systems there are, the dosing requirements, storage requirements, and amenability to mass production, which is critical for the raging pandemic. Here are some, not an exhaustive list of the lead vaccine platforms, but I just wanted to utilize this opportunity to highlight some of the pros and cons of each approach. So towards the left, we have a messenger RNA, which we know is a potent stimulator of the immune system, easy to produce, mass produce, which is an advantage in this setting. However, some of the limitations include, because of the fragile nature of the messenger RNA, that it needs a cold chain, and also the lipid nanoparticle formulation, which forms that protective shell for the messenger RNA, can be one of the reasons why some allergic reactions are occurring. For the viral vector vaccines, they have the advantage of really stimulating robust T-cell responses. However, one limitation are anti-vector immunity. For example, the adenoviruses could face limitations with high anti-vector immunity in African populations, for example. Inactivated viruses tend to be safer than using live attenuated, and you can imagine that's a desirable property for vaccines that you'd wish to use in people with compromised immune systems. However, the production process is quite laborious. Protein subunit vaccines, we have a lot of experience with. They're relatively safe, they're cheap to produce, but they tend to trigger less T-cell responses and require adjuvants for optimal immunogenicity. It's been nice to see that there's been tight coordination between the clinical trial groups as well as regulators like the FDA and agencies within the World Health Organization. And these were critical early on to inform the design, desired outcomes, and approval requirements by these agencies. To the left are some of the guidance around high quality studies that could be conducted. Typically, these would be placebo-controlled, randomized controlled trials, representative of different racial ethnic groups, representative of the populations within a setting that have the disease, with a good sample size to be able to show significant outcomes. Also, with the requirement for a decent duration of follow-up, 2 years, so to adjudicate longer-term safety outcomes as well as durability of vaccines, and importantly, to show efficacy against preventing COVID-19, including severe disease, given some of the concerns of antibody-mediated, antibody-enhanced disease. Similarly, built into the protocols were robust interim assessments for safety boards to assess for efficacy or futility of the intervention, as well as to detect any safety concerns during the conduct of the study. To the right are some of the requirements for emergency authorization or approval, including significantly that satisfactory manufacturing, quality controls, and facilities have to be in place. A desire that vaccines should have greater than 50% efficacy, sufficient, at least minimum 2 months safety data post last dose of vaccine. And the World Health, in addition, had set out prioritization characteristics, including that single-dose vaccines would be preferred over double-dose vaccines, and that also having vaccines that produce at least 6 months durability of protection will be desired characteristics. Now let's talk about the different phases of studies and what we've learned to date. Preclinical studies have been critical and instructive and to inform subsequent human phase trials. For example, studies in mice and non-human primates has allowed evaluation of vaccine antigen constructs to select the best candidates. They've allowed for immunogenicity studies as well as immune profiling, which have been critical to assess those parameters in animal studies, and also have allowed for viral challenge studies that have allowed us to answer important questions, like if it prevents asymptomatic disease or replicating virus. And these have really been critical to inform human trials and provide some very valuable data. Let's talk about safety. So safety assessments have been built in strongly into the vaccine clinical trials protocol. To the right is a figure from the Moderna study, looking at the messenger RNA vaccine approach. We know that for the clinical trials, safety monitoring was built into the protocols so that there were interim reviews to assess for harms that would lead to either triggering a pause or halting clinical trials. And we saw that kick in for a couple of vaccine candidates where there was events that had to be reviewed closely before the trials were allowed to continue. And that should really reassure individuals that safety was a key goal or target for these studies. The studies typically included actively collected data on solicited side effects, which were intensively collected for the first 7 days or so post-vaccination, and also unsolicited adverse events were also captured. Now, the figure on the right shows that after the first vaccination, that individuals could have anything from local reactions at the site of injection, which can be pain, redness, or swelling, and also systemic reactions that could be flu-like symptoms, fevers, chills, fatigue, and arthralgias. Looking at the figure on the right, comparing reported side effects from vaccination 2, you can clearly see that after the second vaccination, that there was more frequency of these events and also increased spectrum of severity. However, the good news is that the majority of these side effects are mild to moderate. The commonest local reaction is injection site pain, and the commonest systemic reactions are flu-like illnesses. The good thing is they occur early, usually within a day or two of injection, and resolve pretty quickly. We found that older individuals tend to have less reactogenicity in these trials. The good news is that there is a very low incidence, generally, of severe adverse events, particularly vaccine-related adverse events supporting decent safety profiles. Let's talk about immunogenicity. Immunogenicity assessments have been critical to assess both humoral and cell-mediated immune responses to vaccination. To date, they have focused on antibodies, the spike protein, which is the vaccine antigen for the vast majority of the vaccine approaches, other than whole viral approaches, or targeting the receptor binding domain. They've also assessed neutralizing antibody titers as well as cell-mediated immunity. If you look at the figures on the right, this is a dose-ranging study for the Pfizer mRNA study that evaluated 10 micrograms, 20 micrograms, and 30 micrograms study. Everyone received 2 doses of vaccine at day 1 and 3 weeks later at day 21. The top 2 boxes represent the antibody levels, IgG, in younger individuals to the left, age 18 to 55, and older individuals 65 to 85 to the right. And the lower boxes, it represents the 50% neutralizing titers, again, in younger individuals to the left, older people to the right. Now, immunogenicity studies have used convalescent sera from healthy individuals as the comparator or the bar for what the target antibody levels and neutralizing titers should be. And this is depicted by the yellow horizontal line on both parts of the figure. So many of the platforms have shown that after the first dose, you don't really achieve that target concentration. However, the second dose acts as a boost, and that's where individuals across the board tend to approach or exceed the target antibody or neutralizing titer levels. So this have shown the importance of second doses as a boost for majority of the vaccine platforms. And longitudinal studies remain ongoing to assess the levels over time. However, an important thing to mention is that the correlates of immunity are still being worked out, and hopefully with phase 3 trials, looking at individuals, for example, whom had breakthrough infections with vaccination and what their antibody levels were, we may learn more about the correlates of immunity. Important to mention again, we saw that older individuals have lower reactogenicity, and similarly, they also have lower neutralizing antibodies across the board. So the figure to the left shows different vaccine approaches, viral vector approaches, inactivated viral vector approaches, and messenger RNA. And you can see in all these vaccines, comparing younger people to older individuals, there's a decreased neutralizing titer for all these vaccine technologies. However, the figure to the top right shows you reassuringly that in spite of the decreased neutralizing titers in older individuals, they remain above and exceed that of convalescent sera titers. And we have seen that, that this is clinically important because in the phase 3 trials, it appears that in spite of what appears to be lower antibody levels or neutralizing titers, it doesn't appear that age is impacting efficacy. Another caveat is that the level of neutralizing titers or antibodies do not always correlate with the degree of immunity against the disease, and there are many immune responses that protect against viral infections that we don't measure with either of these approaches. A question that's frequently asked is what's the durability of the vaccine responses? It remains a work in progress. However, data continues to emerge from the earlier phase trials about the durability of responses following vaccination. This is data from the Moderna messenger RNA study that evaluated trends of antibody levels to the receptor binding domain or pseudovirus neutralization assays over a 120-day period. Now, they found that the binding and neutralizing geometric mean titers exceeded those of convalescent sera, and this was convalescent sera obtained from individuals who are at least a median of 30 days or about a month from the time of their COVID diagnosis. As these studies continue, we will continue to learn more about the durability of protection. And for now, it looks good at 4 months post first vaccination. Let's discuss efficacy. There's been a plethora of reports, especially from the phase 3 trials, on the efficacy of vaccine candidates. In this table, we show the efficacy reports to date, again, favoring published data. The messenger RNA technologies with Moderna reporting a 94% efficacy against symptomatic COVID-19 and Pfizer reporting 95% with the same endpoint. We saw different results from the AstraZeneca study, depending on if you got a standard dose first and a standard dose later, or half dose first and a standard dose later, averaging about 70% efficacy. We saw and heard recently about from the viral vector study, Johnson & Johnson single dose vaccine, about a 66% overall efficacy, as well as from Novavax about their efficacy showing differences by variant. It's clear from studies that were conducted in parts of the world that at the time where there were circulating variants, for example, the variant that was identified in South Africa with the concerning 484 mutation in the spike protein genome, we see that there was, it took a hit on efficacy, both for the Johnson & Johnson vaccine as well as the Novavax vaccine. Good enough is that many of these trials, beyond having met their primary endpoint, are expanding their protocols to groups that were previously left out of initial trials, including a little bit of younger children and adolescents and individuals with certain immune compromising conditions. And we hope and in the future expect that we would have even more data in groups like pregnant women and even younger children. Important to mention that vaccines also have shown protection against asymptomatic infection, which is important from a transmission standpoint. I decided to share this data, which is a preprint from the Lancet regarding the AstraZeneca vaccine. And here they looked at the vaccine efficacy as it relates to the time of administration of the first dose of vaccine. And so in this study, they conducted surveillance for asymptomatic infections by weekly swabs of the nose and throat. So overall, and the bottom line results are that any PCR positive cases of asymptomatic infection were reduced by 67% in individuals even after a single dose of vaccine, suggesting a 2/3 reduction in PCR positive nasal swabs anytime from 22 to 90 days after the first vaccine. A phase 3 study of the Moderna vaccine also showed, even though the numbers were low, 67% reduction in asymptomatic infection as well. Now, this is a model I have created that shows what appears to be the relationship between protection against disease severity and vaccine efficacy. And I think if you look at the most recent or contemporary phase 3 clinical trial reports, it appears there's a graded increase in protection against disease based on disease severity, so that the point estimate for protection against asymptomatic infection tends to be on the lower end, and while you have excellent protection against severe disease and death. However, the protection against moderate to severe disease appears to be influenced by certain things, like the presence or absence of circulating variants that can evade those vaccine-generated immune responses, host characteristics like being immune compromised, and we also need to pay attention to the endpoint definition in clinical trials. However, the finding that the vaccines all have excellent efficacy against severe disease and death are really heartwarming from a public health standpoint. This is a slide that shows the protection that occurred over time in individuals who got at least, these are the 2-dose vaccines. So many of these studies, be it the AstraZeneca vaccine or the Pfizer vaccine, the Sputnik adenoviral vector vaccine or Moderna mRNA study, these studies have always shown, these are the Kaplan-Meier curves of cumulative incidence of COVID-19 in individuals who received placebo and compared to those who got vaccine. And if you look across all the figures presented here, you can see a separation and happening in many instances after the receipt of the first dose and before the second dose, that you already see an efficacy signal in these vaccines. And so we do know that even the first doses of a planned 2-dose series vaccine can confer significant protection against COVID-19. But however, you can clearly see that you need the second dose. And after the second dose, that's typically when the peak efficacy is achieved. There could be a time component to it after receiving the first dose, as we see with the single-dose Johnson & Johnson vaccine, for example, where the protection was measured at about a month after receiving the first dose. Now, the good news is that in general, it appears that certain patient characteristics like age, race, ethnicity, obesity, presence of comorbidities, and prior SARS-CoV-2 infection do not appear to be impacting efficacy, but we will see if these hold on longer-term follow-up. Important to mention clinical trial populations that were included and excluded. We know that for now, most studies have occurred in healthy or non-immune compromised adults. However, clinical trial protocols have been expanded, including the Pfizer study, for example, to HIV-infected patients with well-controlled disease. And also, clinical trial protocols are expanding to even younger children, age 12 and above. However, there remain significant data gaps in kids younger than age 12, pregnant and lactating women, and many categories of immune-compromised patients. So to wrap up the vaccine and clinical trials presentation, just need to remind you that vaccine efficacy appears to improve across the spectrum of disease with the greatest efficacy for preventing severe disease and death. The first dose can provide decent efficacy that exceeds that predetermined 50% threshold, but that the second dose in many of these vaccine platforms boosts immunogenicity and may contribute to durability. We've seen that elderly individuals exhibit lower immunogenicity and reactogenicity, but vaccine protection against COVID-19 appears unaffected. And we know that because we have many effective vaccine candidates available, the era of placebo-controlled vaccine trials, which would allow other vaccine candidates in earlier phases of development to proceed, appears to be closing, if not closed already. Just a few thoughts about vaccine rollout, herd immunity, and challenges to wrap up. Just to mention that achieving herd immunity is usually a composite of the seroprevalence in the community and in addition, vaccine-induced immunity, so that where the seroprevalence is high, you do not need to have that much vaccine coverage. So for example, the estimate is that the UK and US have about a 5 to 20% seroprevalence of COVID-19. And so in that case, you really need just about 50% of the population to receive vaccines to be covered. So we all need to work with these kind of models to help us determine what the optimal vaccine coverage should be. We do know that it is more desirable to get vaccine-mediated immunity than natural immunity because it's more durable. We know that strain-specific immunity would favor vaccines because immunity from prior circulating strains may not hold up against a new variant. And we know that the number of vaccinations needed for vaccines that are less effective would need more. This is another model that assesses or produces evidence on the impact of triaging vaccinations to different groups on transmission and mortality. In this model, they looked at vaccination strategies involving younger individuals, older individuals above the age of 60 in the community, or distributed across all ages. And these are models that are important because of vaccine shortages as they exist. You can model based on the percentage of vaccine supply, the effects on infections and cumulative mortality, and also work out the reductions in infections and deaths over time. So for example, in this model, vaccinating older individuals had the greatest impact on cumulative mortality over time. However, vaccinating younger individuals, okay, had the greatest impact on reduction of infections, as you can see here. So these are critical, and scenarios vary between countries and within countries. And so, models would be really important to inform rollout. Another approach is a risk-based approach, and this is taken from the CDC website that looked at the data for some underlying the comorbidities listed as conferring an increased risk of severe COVID, with the most convincing evidence being those conditions on the left, like cancer and pregnancy, and conditions which could confer increased risk, but for which there's limited evidence, like HIV and liver disease. Having these data are critical to inform prioritization of risk groups for receiving vaccinations. We know that vaccines are currently rolled out globally. As of yesterday, there have been about 168 million doses administered, with an average of almost 6 million doses a day. To the right, you can see the countries listed by their income category, whom have both either confirmed purchases of vaccines or entered agreements to purchase vaccines. In many of these cases, some countries have actually purchased more vaccine than they need for the entire population together. And we can clearly start to see the malavailability or maldistribution of vaccines by income category. Many low middle income countries are yet to start vaccination, unfortunately. In addition to that, logistics for vaccine administration and vaccine hesitancy remain key barriers to optimizing the real-world effectiveness of COVID vaccines, and these will be further elaborated in talks to come. I just wanted to highlight some of the countries that have been top performers and really have been informing real-world outcomes for vaccination strategies. The last slide I will show are some of the approaches to vaccine strategies that can be informed by clinical trials. For example, extending duration of vaccination interval that allows for production of vaccine and administering vaccines to catch up with the demand. To the right is the data from the AstraZeneca study showing that as you delayed the duration, the interval of administering the two-dose strategy, that we actually found an increase in efficacy. The caveat here is that this has only been shown for viral vector vaccines, and we do not have this data for many other vaccine strategies. There are also mix and match approaches that allow you to get one vaccine and get another type subsequently. These are only being conducted as clinical trials. One has been launched in the UK, as is shown below. So let me wrap up this presentation by saying that we have seen that there are multiple phase 3 trials that have shown variable but robust efficacy for many vaccine candidates, especially for the desired outcome of preventing severe disease and death. For most of these strategies, 2 doses are required for optimal efficacy and possibly durability. Safety records have been great. We see that emerging variants do pose a threat, both current and what would be future if the pandemic continues and booster doses are being prevented. And we cannot overemphasize the role of modeling to inform vaccine rollout prioritization, and hopefully we can address equity with vaccine distribution globally to get to the end of the pandemic. Thank you again for having me.